Our next speaker is Maria Jose Endara. She's an Ecuadorian biologist studying the ecology and evolution of plant-insect interactions, including aspects of herbivory, community ecology, chemical ecology, coevolution, and phylogenetics in tropical forests. She's a postdoctoral fellow in the Coley Cursor Lab and an associate researcher at the Universidad Indoamerica in Ecuador. She is presenting today from Quito, Ecuador, where she has been quarantining in her apartment with her young daughter since March. Maria Jose got her BS from the Catholic University in Quito, working on Inga, a tropical tree. In Utah, she has continued to work on Inga and its defenses against herbivores. She has published 15 papers in high-profile journals such as Science and has a first authored paper in PNAS. She's a young fellow of the World Academy of Science, the international equivalent of the US National Academy. And as soon as they can get back into the Amazon, Maria and Lissy Coley will continue their field work on the drivers of speciation in Inga. Maria, are you ready to go?
Yes, I'm ready. Yeah, can I share the screen?
Yeah, you should be able to click on the share screen down at the bottom.
Okay, thank you. Okay, can you all see my screen?
Yes.
Okay, thanks. Okay, good morning, everybody, and thank you, Neil, for the introduction. I'm Maria Jose Endara. I belong to the Coley Cursor Lab, and today I'm going to talk about how the plant defensive traits can regulate the host association of herbivores at the regional scale in tropical forests. Well, it is well known that plants and herbivores are among the most dominant organisms in terrestrial ecosystems. And that's why a key issue in ecology is to understand what are the effects of their interactions on the ecosystem. The evolutionary theory has suggested that the association between plants and herbivores are shaped by the coevolutionary arms race. This theory has also suggested that the main drive of each other's speciation might be the interaction between both groups. And at ecological level, the interactions between plants and herbivores may contribute to the high local diversity of ecosystems, which is a common pattern in tropical forests. So, but what is this coevolutionary arms race? This theory was proposed by Ehrlich and Raven in 1964 and is exemplified in this graph that I'm showing here to the right hand. So in this graph, we have the hypothetical plant phylogeny on the right hand, and on the left side, we have the hypothetical herbivore phylogeny. Sorry, on the opposite. On the right, we have the herbivore phylogeny, and on the left, the plant phylogeny. And the yellow star represents the evolution of a key innovation. So the coevolutionary arms race suggests that the evolution of key innovations, such as glucosinolates in some plant families or latex, has led to the radiation of host plants and the herbivore species. This theory is also a macroevolutionary process, which means that these patterns are expected to be observed at the level of genera or families. So at this level, we will expect to see the evolution of these key innovations. This theory is also called is known as the escape and radiate model. And The principal prediction from this theory is that the association between plants and herbivores is strongly phylogenetically structured, in the sense that closely related species of herbivores are expected to be associated with closely related species of plants. Hence, a pattern of co-diversification is expected, or a high correlation between the phylogenetic histories of both the plants and the herbivores that are attacking the plants. This has been the dominant theory of plant-herbivore interactions in the last 50 years. However, in the last years, several studies have been finding different patterns. One of these patterns, which now is considered as an alternative hypothesis, is the host tracking. So in host tracking, herbivores shift between distantly related hosts. And this is what it has been observed in most of the studies in the last years, which is contrary to what you would expect under the coevolutionary arms race. In fact, what it has been observed is that this shifts between hosts is more associated with how similar the hosts are on their defenses, regardless of phylogenetic relatedness. And this is something that we tested in the Peruvian Amazon at the local scale with the genus of trees called Inga, which is a neotropical genus of trees, and several families of Lepidoptera that were feeding on the leaves of Inga. So here in this graph, I only exemplifying one of the families that we studied in Peru. This is the family called Radiate, which is a family of moths. And when we compare the phylogenetic history for the family of moths Radiate with the phylogeny of Inga hosts, we found no relationship, suggesting that hosts are not shifting among closely related species. But when we compare the phylogeny of Radiate, which is down here on the right side, with how similar were the Inga hosts on their defenses, which is shown here in this dendrogram on the left side, which represents how similar are species on several defensive traits, we found a high correlation between both. Between both. Thus, this suggests that herbivores were actually associated with plants that were more similar on defenses, no matter how related they were. So with the conclusion that we got from this study, and this was at a local site, was that plant defenses play a central role in the evolution of host association. Now, the question was, what happened at bigger regional scales, which is what I've been doing in the last years. So to answer this question, we have been working with data from 5 different sites across Central America and the Amazon, specifically with the genus of trees Inga. This is a picture of Inga, how it looks like. It belongs to the legume family, and it's a very important genus in the Neotropics because it has more than 300 species and is usually one of the most abundant at a given site in tropical forest. So in Panama, we work with 12 Inga species, in Ecuador with 51, in French Guiana with 40, and in Peru with 51 species. At each site, we spent more or less 16 people months collecting data on defensive traits and on the herbivores that were attacking Inga. And for today, for this talk, I'm going to focus on one group of herbivores, one of the most specialized herbivores that are feeding on Inga. They belong to the class Hymenoptera, specifically to the family Argidae, and they are known as sawflies. So as adult sawflies look like this, but the larvae are the ones that are herbivores, which look very similar to caterpillars, but they are not. The studies that we made at the regional level were based on more than 1,200 records, and we identified a host choice for each species using barcodes. basically because at this developmental level, it's very difficult to assign a species name, and also because most of the species are new to science, so we didn't have a way of discriminate between species. The only way was molecular. We also developed the sawfly species tree using 4 markers. We also developed the Inga species tree with 10 nuclear markers. And to estimate how similar were the hosts defensively, we estimated chemical similarity using metabolomics. The questions that we wanted to ask at the regional level were if there's a phylogenetic pattern in Inga defenses, and specifically, and most importantly, what is the pattern that best predict how sawflies and Inga are associated? Is this co-speciation, which will be expected under the coevolutionary arms race? Or is this resource tracking, which is what we found in Peru for Lepidoptera, the graph that I showed you before. And it has been mostly found in several other groups, not only in tropical forests, but also in temperate forests. And also, over what geographical scales have these associations evolved, the associations between Inga and sawflies? We found that the clade of sawflies that is associated with Inga, it's a monophyletic group of very specialized herbivores, and all of them belong to one single family, which is here inside this purple box, the family Argita. The analyses were based on 41 sawfly species, and each sawfly species was associated with 1 or 2 Inga species, suggesting really a very high specialization. Even though at each site you can find like 30 or 40 different Inga species, still sawflies were only associated with 1 or 2, suggesting high specialization. As I mentioned, they are a monophyletic, well-supported clade. And analyses that we performed to estimate the age suggested that the common ancestor for this clade at 6.32 million years. Well, so because we were trying to test the coevolutionary theory at the regional scale, we compared the phylogenetic history for Inga, which is on the left side, with the phylogenetic history for sawflies on the right side, and we found a significant correlation between both phylogenies. We observed that closely related species of sawflies were feeding on closely related species of Inga, or even on geographically separated species of the same Inga host. Like, for example, we have these sister species of sawfly, Motu 32 and Motu 31, which are associated with the same species of Inga, but at different sites, which is Inga alba in Peru and Inga alba in French Guiana. However, this was not always the case. We also found single sawfly species feeding on phylogenetically distant Inga species, such as this sister sawfly species, Motu 37 and Motu 36, one associated with Inga umbratic in Ecuador, and the other associated with Inga uristella in Ecuador too, but species distantly related. And also we found instances where closely related sawflies were feeding on phylogenetically distant Inga species, like this sister species, Motu 8 and 7, each one feeding one on Inga umbellifera in Panama, and the sister species feeding on Inga bourgognei in French Guiana. Hence, even though there is a significant correlation between both phylogenies, something else is going on. Next, what we did was to compare the phylogenetic history of the sawflies with a dendrogram based on chemistry on Inga. And what we found when we ran evolutionary models, when we were able to control for the phylogenetic effects, we found that a very strong effect of defensive chemistry, suggesting that closely related species of sawflies are feeding mostly on species of Inga that are similar in chemistry. When we perform the opposite analysis, the effect of phylogeny controlling for chemistry, yeah, there was an effect, but it was not as stronger as the effect from chemistry. Thus, the association between soft lice and Inga is mainly due to chemistry. And these boxes just exemplify two clades in soft lice that are contributing more to this association, together with these clades of Inga that are also contributing more to this association. Then we have the question, is this a co-diversification or is this a delay resource tracking? If we follow, if we follow the predictions from the coevolutionary theory, we found that yes, both topologies are congruent. So you may say both are co-diversifying. Also, when we compare the ages of the two radiations, Inga has an age of between 4 to 10 million years of radiation, whereas soft lice has an age of between 4 to 6. So you may say that more or less they are congruent. So only based on these two observations, we could suggest that maybe there is coevolution between Inga soft lice and soft lice phylogeny. However, there is also the alternative hypothesis of delay resource tracking, which could suggest that soft lice, like the soft lice ancestor, colonize the clade of Inga that was already that has already radiated and radiate onto the clade. This mechanism will produce the same observation, the same pattern that we are finding, but it does not mean that there was a coevolution between both groups. And the fact that the evolutionary analysis that we perform suggests that chemistry, when controlling for phylogeny, is actually more important on explaining these associations, suggests that maybe delay resource tracking is the most possible, is the best, maybe the best explanation for the observed pattern, but we cannot really know which one is. Now, regardless the patterns of diversification for soft lice, our model suggested that pairs of Inga populations and soft lice species that are occurring are in general occurring in non-overlapping regions. And this will be a pattern consistent with allopatric divergence for soft lice on the same Inga population. For example, one of the examples that we saw in the other slides is like we have this MOTU32 and MOTU31 soft lice. They are associated each one with the same Inga species, but at different site, which is more in agreement with the pattern of allopatric divergence. It could be that the ancestor species soft lice follow the host Inga alba to another country, and because of barriers like distance or geographical barriers, there was no more gene flow and a pattern and a result of divergence between the populations of soft lice happened. And this is a pattern that is seen in more than 60% of the associations between soft lice and phylogeny, which suggests that most of divergence for this group of herbivores might be because of allopatric divergence. Oh, this is another example. Like we have, for example, Inga soft lice, sister species MOTU13 and MOTU11. They are also associated with the same species of Inga, but in different sites. We have, they are associated with Inga lorraina in French Guiana and Inga lorraina in Ecuador. So the conclusions that we have getting so far with these studies at regional level and with soft lice is that it's very important to include traits. In this case, relevant traits such as defenses, which are the traits that herbivores are using to choose hosts. And this is an excellent example um, highlight the importance of including traits. Because if we have only work on the phylogenies of the sawflies and the phylogenies of Inga, we have concluded that there is co-diversification between both groups. But because we were able to include traits and control for phylogeny, we see now that might be something else. It might be another pattern such as delay resource tracking. Another conclusion is that as we are seeing a similar pattern to what we found on Lepidoptera in Peru, and is that closely related species of both sawflies and Lepidoptera are constrained to feed on hosts with similar defenses. We basically have sorry, there's a mistake here. We basically have one mode of diversification, which is allopatric for the majority of cases of sawflies, and that this contradicts the paradigm for herbivore diversification theory. And with that, I conclude the presentation. Thank you.
Thank you, Maria. That was very nice. I think we have time for a couple of questions. Austin, do you want to handle these?
Yeah, of course. Great talk, by the way, Maria Jose. You did great. And so Talia asks, are there estimates for how many secondary metabolites underlie these herbivore specificities? For example, is it only one or a few of the whole cocktail that dictates which plant an insect eats?
Yeah, I didn't show those results here, but we did some analysis specifically to see if there were a specific group of compounds that were more important for sawflies when selecting hosts. And what we found was that a specific kind of compounds called amines were more important for sawflies. And actually, as I mentioned at the beginning of the talk, that so in general, we'll find a big diversity of compounds in Inga. And these specific compounds that I mentioned, the amines, are only present in the species of Inga that are associated with sawflies, which could indicate why sawflies are not feeding on all the Ingas that they're finding in the community.
Very cool. Another question from Bill Andreg was, first off, great talk. Is it known how far sawfly individuals or populations can disperse? And how similar are the sawfly communities across the 5 forest sites?
Yeah, I don't know if there's information about how far they can disperse, but I'm thinking that maybe it's not that far, since we are looking at these patterns of allopatric divergence. The main pattern of divergence can be allopatric. And how similar are the communities? So if at the level of just presence access of the species, the communities are very different because they don't share a species of sawflies. But if we include the phylogenetic information for the communities of the sawflies, we will see they're pretty similar because you find sister species between sites. So yeah, it depends of the scale.
Awesome. Well, with that, if you have any other questions for Maria Jose, please put them in the chat. I'm going to put them all together and send them directly to Maria Jose so that she can answer them as the retreat goes on. So if you have any other questions, please feel free to put them in the chat. And with that, Neil, I'll throw it over to you.
Okay, thanks, Austin. And again, thank you, Maria Jose, for presenting all the way from Ecuador today. That's one of the advantages of doing these meetings over Zoom, is that you can pull people in from all over the world to talk. So well done. Thank you.